Today's lecture is about the first chapter, which is related to the transformation of food into nutrients. Activity 1, our food. Um, let's start by general introduction about food. Food is any substance consumed by an organism for nutritional support, giving energy, helping in growth, important for body repair. So, for example, the lipids and carbohydrates are very important because they give us energy, while proteins are very important for the growth of babies or for kids, and the proteins also are very important during the body repair when the cells multiply and or synthesize new organic matter such as hormones or digestive enzymes, for example. Okay, let's focus here. The food contains organic substances and inorganic substances. So our food is a mixture of two substances, organic and inorganic. What is the difference between organic and inorganic? Organic organic substances are substances mainly composed of carbon atoms. So here in organic substances, the carbon is very essential, while during inorganic substances the carbon is not essential. يعني here in organic substances we found high number of carbon atoms, or sometimes we find a little number of carbon atoms, but always the carbon is essential. Always we found carbon. We cannot found organic substances without carbon. But here in inorganic substances, sometimes the carbon is present, sometimes is not present. Okay, so this is the main difference between organic substances and inorganic substances. Now let's focus on the classes of organic substances. We have carbohydrates, proteins, and lipids. We're gonna talk about each one of them later, or in the next slides. Inorganic substances like the minerals, calcium, iron, sodium, magnesium, okay, and water. Let's start by the first one, the carbohydrates. The carbohydrates are organic substances. We can call them carbs. They are made up of sugar molecules. Bodies break down carbohydrates into sugars that are used to give energy. So the main role of carbohydrates is to give energy, and you have to know the carbohydrates are made up of sugar molecules. Carbohydrates can be classified based on their number of sugars. يعني if we can classify the carbohydrate based on the number of sugars. If the carbohydrate is made up of one ring of sugar, so in this case it's a monosaccharide. So monosaccharides are carbohydrates making up of one ring of sugar only. Okay, disaccharide, they are carbohydrates that are made up of two rings of sugars. So here you have to focus on the number of rings of sugars. If the carbohydrate is made up of one ring of sugar, so in this case it's a monosaccharide. If the carbohydrate is made up of two rings of sugar, so in this case it's a disaccharide. And if it's made up of very high number of rings of sugar, so in this case it's a polysaccharide, and it's more complex. So this one is more complex than disaccharide, and the disaccharide is more complex than monosaccharide. So here the monosaccharide is made up of one ring of sugar, as we said, such as the glucose. Look at this one here. It's a monosaccharide, it's a glucose, and you have to know how to draw a ring of sugar. Okay, this one is made up of one ring of sugar, so it's a monosaccharide. Disaccharide is made up of two rings of sugar, such as the maltose. Look at this example, two rings of sugar, it's clear. And this one بكون في عنا more بعد أكتر وأكتر. Like the starch is made up of very high number of rings of sugar. It's very complicated. So after studying about carbohydrates, now let's study about proteins. Proteins are large complex molecules that are made up of high number of amino acids. So here the proteins are made up of what? of amino acids, not like the not like the carbohydrates that are made up of rings of sugar. So here the proteins are made up of what? Of amino acids. They do most of the work inside a structure and functional roles. It's not important for you now. The amino acids. So here you can see a protein. It's very complicated. Look at the peptides. How many peptides are there here? Three. Three peptides. And look at the amino acids. So amino acids are very simple. When the protein is very complicated or more complex. So proteins is made up of peptides. Peptides are made up of amino acids. And also we can say no, the proteins are made up of amino acids. So proteins أكتر simple بنروح على البيبتايد اللي هني جروب اوف امينو اسيدز صغار، اكتر واكتر سمبل بنروح على الامينو اسيدز بيكونوا فري، ما في حدا عامل بايند مع التاني. So carbohydrates is done, proteins is done. Now let's talk about lipids. Lipids are made up of fatty acids and glycerol. You have to memorize these terms, fatty acids and glycerol, that play an important role to give energy. عندهم كمان أدوار تانية. This is a molecule of lipid, and the molecule of lipid after its digestion, it gives one glycerol and three fatty acids. So look at the lipid of the molecule of lipid is made up of one glycerol linked to three fatty acids. Okay, so carbohydrates are made up of rings of sugars, proteins are made up of amino acids, and lipids are made up of fatty acids and glycerol. Minerals are found in our food, for example, calcium, phosphorus, aluminum, sodium. They are different. They have different functions in muscles, in bones, in nerve impulse, etc. Here, the insulin, it's a hormone made up of amino acids. So here are amino acids. Okay, so it's a protein since it's made up of high number of amino acids. The starch here is made up of high number of rings of sugars, so it's a carbohydrate and it's a polysaccharide because it's made up of a high number of rings of sugars. And this one is a lipid because it's made up of three fatty acids and one glycerol. This one is maltose, which is disaccharide carbohydrate because it's made up of two rings of sugars. As you see here, it's given two rings of sugars for Azanaida, maltose. The glucose is a carbohydrate made up of one ring of sugar, so it's a monosaccharide. And this one is a protein since it's made up of amino acids. Look at the given here, it's a amino acid, so it's made up of amino acids. فمنقول له هيدا it's a protein. Food tests are qualitative tests and not quantitative, so we can determine the quality. We can determine if the starch is present or not. We can determine if the protein is present or not. We can determine if the lipids are present or not, but we cannot determine the quantity. We cannot determine the quantity of starch in your food. You cannot determine the quantity of proteins in your food. So they can determine if it's present or no. They are used to test some substances in any food, such as iodine test, Fahrenheit test, and Biuret test. Let's start studying with the first test, which is the iodine test. The iodine test is used to check the presence of starch. So here we can determine the presence of starch. It's a test done without heating, no need to heat. It's very simple, as you see here. In the presence of starch, the brown orange of the iodine solution becomes dark blue. So here we have a potato. We are testing the presence of starch inside it. Here the iodine solution. The color of the iodine solution is brown orange, as you see here. After adding some drops of iodine solution, this color becomes dark blue, indicating the presence of starch. So you have to memorize very well the color, which it changes from brown orange into dark blue in the presence of starch. So here the results of iodine test done with two samples. Here it's negative because we have no dark blue color, and here is positive, indicating the presence of starch. So this sample contains starch and this sample doesn't contain starch. We cannot determine the quantity, by the way, of starch here. We can determine only if the starch is present or not. Here the Biuret test. The Biuret test is a test used to check the presence of proteins. in a sample. So here it's a Biuret test which used to check the presence of proteins in a sample by adding what and what? Copper sulfate, then sodium hydroxide. So here in this test, we need to add two substances. You have to memorize them. We need to add copper sulfate then sodium hydroxide to a sample. As you see here بتشوفوا انه we add copper sulfate then we add sodium hydroxide بيطلع الكالر blue. In the presence of proteins بيرجع بصير violet. So this test, Biuret test, is done to check the presence of proteins. We need to add copper sulfate and sodium hydroxide, and in the presence of proteins, the blue color, here I have a mistake. The blue color turns into violet color, indicating the presence of proteins. So do we need here any heating? No, we need no heating. We don't need heating here to do this test. It's a simple test. Just we need to add copper sulfate and sodium hydroxide, which has a blue color, then turns into violet in the presence of proteins. So here blue color, which indicate the absence of proteins, and plus, which mean indicate the presence of proteins. Also, you have to focus on this the Biuret test. The Biuret test كمان also gives a positive color with violet color with proteins and peptides. يعني إذا أنا معي proteins أو معي peptides, both of them give the same color which is violet color. يعني ما بقدر فرق إذا معي proteins and peptides بقلب التيوبز لأنه اتنيناتهم حيعطوا the same result. هون I have to do coagulation test. فإذا ليكو هون proteins I have to start by coagulation test. Coagulation test هو when we heat under any sample. يعني بسخن تحت منهم، إذا جمد مثل البيض ببين إنه عندي بروتينز، بس البيبتايدز ما بجمدوا. يعني بروتينز بيقدروا يعملوا coagulation after heating، بس بيبتايدز ما بجمدوا بس أعملهم heating. فcoagulation test مع البروتينز بيطلع positive، مع البيبتايدز negative، مع الامينو acids negative. فإذا if I have 3 tubes, one of them contains proteins, the second one contains peptides, and the third one contains amino acids. Proteins after coagulation يعني بعملهم heating بيطلعوا positive، peptides بعملهم heating بيطلع negative، amino acids بيطلع negative. no coagulation، لو سخنت ما حيطلع. بينما if I do Biuret test, proteins and peptides give what? the same result اللي هي violet. فكيف بقدر فرق بين البروتينز والبيبتايدز باي coagulation test؟ كيف بقدر فرق بين proteins والامينو acids او peptides وامينو acids باي Biuret test. So we can distinguish between proteins and from peptides by coagulation test because they give the same result with Biuret test. وامينو acids بستعمل Biuret. هلا تيستينج ليبيدز كتير سهل. بعمل رابينج او بدق اون كراش سامبل فود بيعطي ترانسلوسنت سبوت بطايلس، يعني بتلاقوا هون على طول بتضلها مطايلسه. Fahling test هو a test which is done to check the presence of reducing sugars, and here we need heating. okay. فإذا Fahling test هو to check the presence of reducing sugars. Fahling test identifies the presence of reducing sugar. It's performed with heating. بدي أعمل heating. In the presence of reducing sugars, the blue color is turned into brick red precipitate. فإذا شو حيعطي؟ brick red precipitate. بيكون blue بتحول into brick red precipitate. حنشوف هون as you see here we need what? heating. Fahling test مثلا عطى blue at the initial. هون كلهم عطوا blue بالأول، و after at the end of Fahling test هون we have a positive because of the presence of brick red precipitate, negative and negative. فإذا which one of them is a reducing sugar? The glucose is a reducing sugar. فإذا this one, the glucose اللي هو monosaccharide which is made up of one ring of sugar is a reducing sugar. But the sucrose is not a reducing sugar. Why? Because the color stays blue. And starch which is made up of high number of rings of sugars هو بعد حيكون فيه أكتر بعد هون is a non-reducing sugar. فإذا sucrose non-reducing sugar وهيدا ما حيعطي only glucose is a reducing sugar. لأنه عطاني بريكرات بريسبتيت after performing فهلينج تيست. فإذا هون عندي مونوسكرايد دايسكرايد بوليسكرايد. هون ال color of فهلينج test changes from blue into brick red precipitate، يعني هيدا reducing sugar. from blue stays blue منه reducing sugar، blue stays blue منه reducing sugar. Here in figure B page 16 we are testing the starch in a bread. So we add a few drops of iodine solution. The color turns into dark blue, from brown orange into dark blue, indicating the presence of starch. And here we are performing biuret test because we are adding copper sulfate than sodium hydroxide. So in this case we are performing or we are carrying out a biuret test to check the presence of proteins which is present in the bread, because the blue color turns into violet. So from blue into violet معناتها في proteins. From brown orange into dark blue معناتها في عندي starch. فإذا the addition of iodine solution to a slice of bread gives dark blue color, indicating the presence of starch. And the addition of copper sulfate than sodium hydroxide to a slice of bread gives violet color, indicating the presence of proteins. Thus فإذا شو بقلبه البريد؟ بقلبه starch and proteins، وفي يقول بقلبه carbohydrate اللي هو starch and proteins كمان. correct. Here we are testing some inorganic substances found in bread. هون حطينا شقفة خبزة، سخنا، عملنا لها heating، عطت few drops of water in the inner wall of the tube. فبهالحالة فينا نتأكد انه شو عنا، انه عنا water inside the bread. هون زدنا distilled water وعطينا few drops of silver nitrate. فإذا here we are testing the presence of salt اللي هو sodium chloride. فإذا عم شوف إذا البريد بقلبه ملح أو لا. في عنا many steps. أول step we have to add distilled water. Then we add to add few drops of silver nitrate. لا، تاني step تكون shaking it well. تالت step هي اللي adding few drops of silver nitrate. in case عطتني white precipitate معناتها في عندي salt. in case ما عطت white precipitate no salt. فإذا what are the steps to check the presence of salt in any food? First step, you have to add distilled water to your food, to the tested food. Then we have to shake it very 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 well. بحركها كتير كتير منيح. After that we have to add few drops of a solution of silver nitrate. In case we obtain white precipitate معناتها في عنا salt, indicating the presence of salt. in case ما عطى فإذا no salt. فهون أكيد عندي salt لأنه it gives white precipitate. فإذا شو هيدا البريد؟ شو بقلبه؟ البريد بقلبها water وبقلبها salt. فإذا بقرأهم هون بفهمهم كتير واضحين. فإذا ليش هو البريد is a complex substance؟ لأنه عنده starch، لأنه عنده proteins، لأنه عنده water وعنده salt. يعني how many substances اكتشفنا لهلا بقلب البريد؟ مثل ما شفنا هون في عنده starch، مثل ما شفنا هون في عنده proteins، مثل ما شفنا هون عنده water، مثل ما شفنا هون عنده salt. يعني كم substances بقلبه؟ 4. فإذا هو a complex food، لأنه عطول نحن الفود هو complex بيتألف من many substances. هون grapes عم نعمل لها iodine test. اوكي، حنشوف انه عطت براون اورنج يعني no starch. البنانا مع الايودين تيست عطت dark blue presence of starch. Potatoes with iodine test gives positive, indicating the presence of starch. Fish with iodine test عطت no dark blue color, absence of starch. Now failing test هو which needs heating. هيدا to check the presence of reducing sugars. عطينا brick red precipitate, indicating the presence of reducing sugar, no reducing sugar, no reducing sugar, no reducing sugar, because the color obtained is blue, and here is black red precipitate. Biuret test is a test to check the presence of proteins. In grapes عطا blue ما في. with banana عطا blue negative. mashed potatoes يعني مع عصر بطاطا وحطوها كانت تطلع negative. mashed fish عطت positive. violet يعني violet, indicating the presence of proteins. فإذا here we have to add copper sulfate and sodium hydroxide. فإذا مثل ما قلت كله واضح. يعطيكم العافية.